Welcome back to this next video in which we are discussing atherosclerosis. Okay, so let's summarize where we've got to so far with the pathogenesis of atherosclerosis. So we are looking at the response to injury model of atherosclerosis, which is a very widely accepted model for the pathogenesis of atherosclerosis. And in this model, the starting point for the pathogenesis of atherosclerosis is that you are going to get chronic injury occurring to the endothelial cells over the area where an atherosclerotic plaque is going to form. Okay, and this chronic injury could be due to one of these four factors that we've listed here. So hypertension, too high blood pressure, hyperglycemia, too high blood glucose concentration, such as would occur with poorly controlled diabetes, the toxins of cigarette smoke, which will be present regularly in the blood if you're smoking regularly, and also turbulent blood flow such as occurs at the junctions of blood vessels. Okay, so these things can all cause chronic damage to the endothelial cells, and the idea then is that the formation of an atherosclerotic plaque is due to these endothelial cells dysfunctioning because of this injury and responding as though the way they would if they were endothelial cells in capillaries at areas that were infected, for instance, with bacteria. Okay, so they're going to start recruiting white blood cells from the bloodstream into the subendothelial space, and they're also going to increase the vascular permeability. So they bring in macrophages into the subendothelial space, and they bring in T lymphocytes, two very powerful cells of the immune system. Also, the increase in vascular permeability results in LDL particles from the bloodstream, which we've discussed are these floating stores of cholesterol that you have within your blood. They're also going to end up in the subendothelial space. And what we now want to discuss is how these things coming into the subendothelial space is going to then lead to the formation of an atherosclerotic plaque. Okay, so we need to go now to the macrophages once again. So the macrophages, remember, they are a cell of the immune system. They have been called into the subendothelial space by these dysfunctioning endothelial cells, and they believe that there is some sort of pathogen in the subendothelial space. That's the reason they're recruited to an area, to destroy pathogens. So they have now come into the subendothelial space with the intention of wielding all their weapons and destroying pathogens. Okay, so what they're going to do is they're going to release a bunch of truly horrible molecules. So one of the tactics that macrophages have for killing bugs is to release a bunch of incredibly horrible toxic molecules that will react with the bacterial proteins and the bacterial membranes, all of the molecules that make up a bacteria, and will damage the bacterium. So basically the macrophages are going to release molecules, very reactive chemical molecules, that are going to poison the bacteria. Okay, and these are going to be important in the next step of the pathogenesis of atherosclerosis. So we can summarize these horrible molecules by ROS, which stands for reactive oxygen species. So ROS here stands for reactive oxygen species, and these are molecules which contain oxygen atoms and which are incredibly reactive and toxic to bacterial cells. Well, not just bacterial cells, to human cells as well, but the idea is we've got a bacterial infection here. Yes, if we damage a few human cells, that's tragic, but we need to get rid of the bacterial infection. So it's a nasty weapon, but one that we're going to have to use here. So reactive oxygen species is what ROS stands for. Okay, so let me give you some examples then of these reactive oxygen species, these horrible molecules with oxygen in. Okay, so one good example is hydrogen peroxide, which is H2O2. So this is hydrogen peroxide, so water with an extra oxygen in it. Okay, so let me draw out the structure then for hydrogen peroxide, and this has a peroxide bond in, and the peroxide bond is when you have an oxygen bound to another oxygen with a single bond. So this bond here is known as the peroxide bond. Okay, and then of course we'll have 2 hydrogens bound as well, one off each of these oxygens, like so. So here is hydrogen peroxide, a very reactive molecule which will attack bacterial cells. Another good example of a reactive oxygen species is superoxide, the superoxide free radical. Okay, it's an ion and a free radical. So let me just explain the structure of superoxide. So this is called superoxide. So superoxide is what you get from oxygen if you fire an electron at oxygen. So let me draw out oxygen here, and I'll draw out the electronic dot and cross diagram for oxygen. So this is the normal oxygen molecule, dioxygen, where you have an oxygen atom bound to another oxygen atom by a very stable double bond. This is the sort of oxygen that, you know, is all around us in the atmosphere. Okay, so let me put the electrons on here. So each of these double bonds here will consist of 2 electrons, like so. So the crosses can be from this oxygen here, and this oxygen will also have 2 lone pairs of electrons, and the dots will be from this oxygen here, and this oxygen will also have 2 lone pairs of electrons. Okay, so this is the normal stable dioxygen molecule. Now what would happen if we fired and made this oxygen molecule accept another electron? So we're going to fire an additional electron in here. So we'll have this very bad electron, which I will colour in red. Okay, it's a very naughty electron. Okay, it's going to come in at this dioxygen molecule, and what's now going to happen? Well, one of these bonds in the double bond is going to break. And what you can imagine happening is one electron can go back to each of the oxygens. So break one of the bonds, leave one of the bonds intact, but break one of the bonds, send an electron back to each of the oxygen atoms, and then this electron that has now joined in, it will pair with one of the electrons of one of the oxygen atoms. So what we'll end up with is this then now. So here are 2 oxygen atoms still linked by a single bond now. Okay, let's say the electron went to this oxygen here. So that means that this oxygen over here is still going to have 2 lone pairs of electrons, but now it's got an unpaired electron here. And this oxygen over here, it's now going to have 3 lone pairs of electrons because it got this red electron here. So I'll just colour that one in red. So one side of the molecule then is going to have a negative charge, so this is this side over here where the additional electron is, and the other side of the molecule has an unpaired electron, a free radical. Unpaired electrons are extremely reactive. Electrons do not like being unpaired, so this molecule is made extremely reactive by the free radical. So we write O2 negative dot rather. The negative is to show the negative charge on one side of the molecule, the dot is to show the free radical on the other side of the molecule. So this is an incredibly reactive oxygen species. So that's superoxide and hydrogen peroxide. They are 2 major examples of reactive oxygen species. So macrophages will start producing these reactive oxygen species and dishing them out into the extracellular fluid, in the belief, of course, that there is a pathogen present which needs to be annihilated. Now, of course, these molecules are going to do a lot of damage, and particularly relevant to us is what they're going to do to the LDL molecules that have come in. So we know that LDL particles have come from the bloodstream into the subendothelial space because they came through those holes between the endothelial cells, which have come about because permeability of the endothelial cells is increased for these dysfunctional endothelial cells. Now what's going to happen is the LDL particles are going to be exposed to these incredibly reactive oxygen species here. And those reactive oxygen species are going to react with the molecules in LDL and turn these LDL particles into what we now call oxidized LDL. Okay, so oxidized LDL, people often ask, what is oxidized LDL? Well, we actually don't really know what it is that has actually changed in oxidized LDL. All our definition of oxidized LDL is that it's LDL once it's been exposed to these incredibly reactive oxygen species, and these incredibly reactive oxygen species must react with many of the molecules in the LDL and change it in some way, and now the properties of this oxidized LDL, this LDL that's been exposed to reactive oxygen species, are very different. So this is what's now known as oxidized LDL, ox for oxidized, so this is short for oxidized, and this just means LDL that has reacted with reactive oxygen species, and its properties are hugely changed after this, and its properties are going to be pro-atherosclerotic. Okay, so here is the apolipoprotein B100, and just to show that this has changed, I'll put some green sparks around it. So something about this has changed, and it's now going to, as we'll see, be phagocytosed or engulfed by macrophages, and it's going to result in the conversion of those macrophages into something called foam cells. Okay, so as I say, people often ask, what is oxidized LDL? But the definition of oxidized LDL is that it's just LDL that has reacted with reactive oxygen species, and something has changed. What has actually changed that leads to the ridiculously different properties of the oxidized LDL? We don't know. That's still unknown. But something has changed about this once it was exposed to these reactive oxygen species. Okay, right, so the LDL that is in the subendothelial space is going to be exposed to the reactive oxygen species, and it's now going to change properties and be referred to as oxidized LDL. And oxidized LDL, the macrophages don't like the oxidized LDL. They see it as something very dangerous, okay, and they start actually taking it all up. They start trying to get rid of it from the subendothelial space. They've almost interpreted this as the pathogen that they're trying to destroy. Okay, and what happens is they take up the oxidized LDL, and they end up having to store all of the huge number of cholesterol molecules that they get from taking up the oxidized LDL inside their cytoplasm. Okay, so the macrophages get fatter and fatter and fatter, filling up with cholesterol stores, and they they change properties once they've done this, because having a huge amount of cholesterol in your cytoplasm does tend to change the way your cell biology functions. So they change properties, and they're now referred to as foam cells. So I think we'll have to go over the page, unfortunately, and draw a new picture. And in fact, because we've got so little space there, I'm going to get a new piece of paper, and we'll start our new picture on a fresh piece of paper. Okay, right, so here then I will draw our next picture. So bear with me a moment whilst I just copy out the basics. So here, this is going to be the basement membrane, like so. Okay, then we're going to have our endothelial cells sitting on top of the basement membrane. Here's the nucleus of this endothelial cell. Here is another endothelial cell also sitting on the basement membrane there. Right, and now what has happened, remember, is we've had the macrophages and the T cells come in, we've had the LDL come in, the macrophages have started releasing these reactive oxygen species, which have now converted the oxidized LDL, the LDL into oxidized LDL. The macrophages are now going to take up the oxidized LDL, and they're going to convert into structures known as foam cells, and let me draw you a foam cell. Okay, so they are these massive great cells because they're swollen with all the cholesterol that they've got stored in them. Okay, and the reason they're called foam cells is that they appear foamy when you look at them histologically, and the reason they appear foamy is they have all of these huge stores of cholesterol in their cytoplasm. So every single one of these blobs that I'm drawing here, this is a store of cholesterol, a deposit of cholesterol inside their cytoplasm, and when they're actually stained for a histological slide, all of the lipid is washed out, so you lose all the cholesterol. So these blobs end up being white, and it makes them appear foamy. Okay, so here then, these are all stores of cholesterol then. Okay, so I'll colour them in yellow here. Right, so this is now called a foam cell, and this is going to be one of the principal things that is going to be in the lipid core of an atherosclerotic plaque. Okay, so this is a foam cell. Okay, so what's going to happen then over time is you're going to get more and more macrophages coming into the subendothelial space, more and more LDL coming into the subendothelial space. The macrophages will be releasing the reactive oxygen species, converting the LDL into oxidized LDL, and they'll then take up the oxidized LDL and convert into foam cells, and that's what's going to form a big part, the lipid core of the atherosclerotic plaque. Okay, you're just going to accumulate more and more of the foam cells in the subendothelial space, and you can imagine as these things accumulate, it is going to push the basement membrane upwards into the lumen of the blood vessel, and you're going to actually start to form an atherosclerotic plaque. Okay, so foam cells accumulating is one of the major components of the atherosclerotic plaque. Right, in addition, what happens is foam cells, they're not particularly stable cells. Okay, this is not a healthy cell with this huge amount of cholesterol stored in its cytoplasm. So they have a tendency to die, either by apoptosis or potentially even by necrosis. And when they die, the lipid doesn't just vanish. Instead, what happens is all the lipids released into the extracellular fluid. So what tends to happen is you end up with huge, great extracellular stores of lipid, where all the cholesterol is aggregated together because it's all lipid soluble. So you end up with a massive, great extracellular store of cholesterol. So that's what this is supposed to be, a cholesterol droplet, an extracellular cholesterol droplet. So amongst the foam cells in this forming atherosclerotic plaque, you're also going to have extracellular stores of cholesterol, which are formed because foam cells have died and all the cholesterol that was stored in them has then just been released into the extracellular fluid, and it's all aggregated together into a great big extracellular droplet like so. Okay, so those are two principal components then of an atherosclerotic plaque. Loads of foam cells in the subendothelial space and these extracellular droplets of cholesterol that are formed because the foam cells have given in. Okay, but there is another component to an atherosclerotic plaque. The two principal components are the lipid core, and this is what the lipid core consists of, the foam cells and the extracellular cholesterol deposits. Okay, so I'll put this key piece of terminology, the lipid core of an atherosclerotic plaque. And then the other key component of an atherosclerotic plaque is the fibrous cap. And I want to now show you how the fibrous cap is going to form. So over the top of the lipid core of an atherosclerotic plaque, you're going to have a very tough, fibrous tissue, okay, connective tissue that will cover the lipid core and actually protects the plaque, stabilizes the plaque, as we'll see. This is known as the fibrous cap, and we need to see how this is going to form, and this is going to be formed concurrently with the lipid core of the atherosclerotic plaque. So what is it that's going to lead to the formation of this fibrous cap over the lipid core then? Well, we haven't seen the function yet of those T cells that have come in, so let's now see the role that the T cells are actually going to play in this atherosclerosis story, pathogenesis. Okay, right. So I'll just draw back on these macrophages and T cells coming in, because actually both the macrophages and the T cells are going to play a role in this. Okay, so here we've got a macrophage before it's actually converted into a foam cell. Okay, so a macrophage that is fresh and has just come in and hasn't yet converted into a foam cell. So here is our macrophage in red, and then the little cell there, which I'll have in orange. This is supposed to be the T cell that's also in the subendothelial space where our atherosclerotic plaque is forming. So this is the T cell, and this is the macrophage. So these are inflammatory cells. Now, one of the key things that inflammatory cells often do is they release loads of molecules that change the behavior of other cells. Okay, and that's one of the things that these macrophages and these T cells that have come into the subendothelial space are now going to start doing. So they're going to start releasing loads of molecules which we can collectively group together under the umbrella term cytokines. So these macrophages and these T cells come in and they start releasing loads of molecules which are together referred to as cytokines. So what is a cytokine? A cytokine is a molecule that is released by a cell of the immune system, okay, and which acts on another cell, and it doesn't necessarily have to be a cell of the immune system, okay, it has to be released by a cell of the immune system, such as a macrophage or a T cell, but it's going to act on just another cell that might not necessarily be a cell of the immune system, and it's going to change the behavior of that other cell. So it's just signaling molecules that are released by immune cells. That is all the concept of a cytokine is. Now, if you like, you can go away and pick up some pathology textbook, and you can find a great long list of all of the cytokines, all of the specific names of the cytokines that these macrophages and T cells that come into the subendothelial space in the pathogenesis of atherosclerosis actually release. I would advise you, however, not to bother. I don't think for a basic understanding of atherosclerosis, it is necessary, or that it enhances your understanding, to actually know the great big long list of specific cytokines that are actually released by macrophages and T cells in the subendothelial space in the pathogenesis of atherosclerosis. For that reason, we are just going to summarize them all together under this umbrella term of cytokines. So the macrophages and the T cells that have come into the subendothelial space in the pathogenesis of atherosclerosis are now going to release these molecules, which we're going to group together as cytokines, and these are now going to affect the behavior of other cells, and they're going to affect the behavior specifically of smooth muscle cells in the tunica media underneath the subendothelial space. Well, of course, there is the internal elastic lamina separating them. We'll come on to that in a moment. But they're going to go and act on smooth muscle cells, which I will add on to the picture here, and they're going to change their behavior. And the change in the behavior of the smooth muscle cells is what's going to actually generate the fibrous cap of the atherosclerotic plaque. Okay, so let me firstly just add a few more things onto my picture here. So firstly, let's have the internal elastic lamina, shown here in blue. Okay, so I'll just label this up. So this is the internal elastic lamina, the last portion of tunica media. So this is internal elastic lamina. Okay, and then outside of the internal elastic lamina, we of course have the tunica media, which will contain vascular smooth muscle cells. So I'll just draw two of these spindle-shaped vascular smooth muscle cells. So here is this one's nucleus, and here is this one's nucleus. So these are vascular smooth muscle cells, VSMCs for short. So V is for vascular, S for smooth, M for muscle, and C for cells. Okay, so I'll just colour these in red so that they convincingly look like vascular smooth muscle cells. And now the cytokines then have been released by the T cells and the macrophages in the subendothelial space. These are going to go down and act on receptors that are on the surface of the vascular smooth muscle cells, and they are going to change the behaviour of those vascular smooth muscle cells underneath this place where the atherosclerotic lesion is starting to form. And what these vascular smooth muscle cells are going to do in response to these cytokines is they're going to start migrating across the internal elastic lamina, which is certainly a behaviour that they should not be doing. So they're migrating across the internal elastic lamina here into the subendothelial space, and when they get into the subendothelial space, they start proliferating. Okay, so this one will come in, and it's going to proliferate, and it's shrunk a little bit, but of course that's just for the purposes of the picture because I'm running out of space, and it will start proliferating here. So you'll get lots of these vascular smooth muscle cells now in the subendothelial space. And the vascular smooth muscle cells can now do two things to contribute to the development of the atherosclerotic plaque. They can either contribute to the development of the lipid core, or they can contribute to the development of this other portion that we're trying to understand at the moment, which is the fibrous cap. Now, so if the vascular smooth muscle cell is going to become part of the lipid core, then it does the same thing as the macrophages did with regards to oxidized LDL. Okay, so we've got lots of oxidized LDL in the subendothelial space thanks to the macrophages which have released the reactive oxygen species and converted the LDL into oxidized LDL. And now vascular smooth muscle cells, they also want for some reason to take up this oxidized LDL, so I'm just putting those green sparks around the oxidized LDL to remind us that it has special properties. Okay, so the vascular smooth muscle cells, they can end up taking up the oxidized LDL, this LDL that has reacted with reactive oxygen species, and they end up then accumulating these huge deposits of cholesterol in their cytoplasm as well. And whenever any cell accumulates a huge amount of cholesterol in its cytoplasm, it starts behaving in the same way. So the cholesterol changes the behaviour of the cell in the same way. So they then end up being foam cells, just like those that came from the macrophages. Okay, so vascular smooth muscle cells then can also take up oxidized LDL and transform into foam cells. However, some of the other vascular smooth muscle cells that you've now got in the subendothelial space, these might go up to the top of where this lipid core of the atherosclerotic plaque is forming, so they might go up just beneath where the basement membrane is, and they then take on behaviour that is very like fibroblasts. Okay, so they adopt what is called fibroblast-like behaviour. Now, what is a fibroblast? A fibroblast, remember, is one of these cells which makes a lot of connective tissue. It synthesises the extracellular matrix. Okay, so these vascular smooth muscle cells are going to go up to the underside of the basement membrane. They're going to convert their behaviour to this fibroblast-like behaviour, and they're going to start synthesising connective tissue in the way a fibroblast would. Okay, and they're going to synthesise loads of connective tissue and make a very thick layer of connective tissue over the top of the lipid core of the atherosclerotic plaque. And this very thick layer of connective tissue containing the vascular smooth muscle cells that now have fibroblast-like properties is going to be called the fibrous cap. Okay, so what I now want to do then is finally draw out what an atherosclerotic plaque is going to look like, emphasising these two different layers, the fibrous cap and the lipid core. Okay, so firstly, it's best to start by drawing the internal elastic lamina here, because that's going to be below, and the atherosclerotic plaque doesn't extend beyond the internal elastic lamina. So here is the internal elastic lamina. Now let's have the endothelium bulging out into the lumen because of our atherosclerotic plaque now. So this is going to be the basement membrane on which the endothelial cells will be sat. So that's there in green. Okay, and then we'll have the endothelial cells which have been chronically injured and have started this entire process to create this atherosclerotic plaque on the top here. So there are their nuclei. And now, in the space in between the basement membrane and the internal elastic lamina, which is of course the subendothelial space, we're going to have this atherosclerotic plaque, or atheroma. Okay, and it's going to consist of this fibrous cap, which I'll add on firstly, okay, which is this layer of fibrous connective tissue, a very thick layer of fibrous connective tissue, with the vascular smooth muscle cells, which now have fibroblast-like behaviour and are synthesising lots of connective tissue. Okay, so these are these modified vascular smooth muscle cells which were acted upon by the cytokines released by the T cells and the macrophages, and which migrated from the tunica media into the subendothelial space, and which didn't become foam cells, instead took on the fibroblast-like behaviour and started synthesising a huge amount of connective tissue to synthesise this fibrous cap. So all of this portion that I'm highlighting in blue here, this is all connective tissue, and this is the fibrous cap of the atherosclerotic plaque. So this portion here, I'll put a label to it, this is the fibrous cap. Okay? And then underneath what we're going to have is then the lipid core of the atherosclerotic plaque, which is going to consist of loads of foam cells which are derived from macrophages which have taken up oxidized LDL, and also vascular smooth muscle cells which have taken up oxidized LDL. And then amongst the foam cells we're going to have the extracellular deposits of cholesterol which have come about from the dying foam cells. So let me now put some of these foam cells on here. So these blobs are going to represent the foam cells, like so. Okay, and I'll give them their nuclei here and colour them in yellow to represent the fact that they are full of this lipid. Okay, like so. And then amongst the foam cells, as I say, there's going to be these extracellular deposits of cholesterol, which I'll also colour in yellow. So this is going to be an extracellular deposit of cholesterol. Let's say this is also an extracellular deposit of cholesterol. Here is also an extracellular deposit of cholesterol, and here's another extracellular deposit of cholesterol, which have formed because the foam cells have given in. Okay, so this portion of the atherosclerotic plaque then, this is what would be called the lipid core of the atherosclerotic plaque. And there is the structure of an atherosclerotic plaque. So we've now completed the pathogenesis of an atherosclerotic plaque. So let's just summarise the entire process, and then I'll say a few more words about atherosclerotic plaques before we have a break and then move on to anti-atherosclerotic drugs. Okay, so the process begins then, we believe, with chronic injury to the endothelial cells, potentially because of hypertension, hyperglycemia, smoking, or because of turbulent blood flow, for instance, at the junctions of blood vessels. Okay, the endothelial cells are chronically damaged, and they then take on this dysfunctional behaviour where they start recruiting cells of the immune system into the subendothelial space, and they also increase their vascular permeability, allowing LDL into the subendothelial space. You end up with macrophages in the subendothelial space, T cells in the subendothelial space, and also LDL in the subendothelial space. And by the way, dotted around the place in the atherosclerotic lesion, you'll also see T cells. Okay, so if you were looking at this histologically, particularly in the fibrous cap, you find the T cells. Okay, so you will see the T cells that are left over and which have helped to coordinate this, the formation of this. Okay, so then what happens, of course, is the macrophages start releasing their horrible molecules, the reactive oxygen species, such as hydrogen peroxide and superoxide. These then react with the LDL particles in ways that we still don't understand and change the property of those LDL particles. And these LDL particles with these changed properties we refer to as oxidized LDL particles. And one of the key new properties that these oxidized LDL particles have is that macrophages and also vascular smooth muscle cells love to take them up. They end up with huge stores of cholesterol in their cytoplasm, and they convert into these foam cells, which are going to form the bulk of the lipid core of an atherosclerotic plaque. So indeed, the macrophages are going to do this. They're going to start taking up oxidized LDL, and they will convert into foam cells. So you'll gradually bring in more and more macrophages, more and more LDL. The LDL will get converted into oxidized LDL, and you will therefore form more and more foam cells, so the lipid core of the atherosclerotic plaque will grow and grow over time. In addition, we've talked about how the cytokines released by macrophages and T cells are going to act on the vascular smooth muscle cells underneath this developing atherosclerotic lesion, and they are going to change the behaviour of the vascular smooth muscle cells to make them migrate from the tunica media into the subendothelial space. They will then divide in the subendothelial space, and their progeny can go on to do one of two things. They can either convert into foam cells by taking up lots of oxidized LDL, or they can go on to form the fibrous cap, so they go underneath the basement membrane nearby where the T cells who are summoning them with the cytokines are, and they'll start taking on behaviour like fibroblasts. Okay, and they'll start synthesizing a huge amount of connective tissue, and they'll produce this fibrous cap over the lipid core of the atherosclerotic plaque. So those are the two components, the two major components of an atherosclerotic plaque. The lipid core made up of these foam cells and these extracellular deposits of cholesterol, which we know come about because the foam cells aren't very stable cells and end up dying and releasing all their cholesterol everywhere. And then also on top of that, protecting it, keeping it encased, you've got the fibrous cap. And this structure can get bigger and bigger and bigger, and clearly it can obstruct the blood vessel. It can lower the diameter of the lumen and therefore damage blood flow. And this is how it can lead to ischemic heart disease if it occurs in the coronary circulation, and how it can lead to, for instance, peripheral vascular disease when it occurs in arteries in the legs. Okay, but as I say, we're not going to discuss in detail the consequences of atherosclerosis occurring at specific sites around the body. Those are huge topics, and they'll be covered in separate videos. Okay, right, the final thing I want to talk about in this video is the concept of a stable atherosclerotic plaque versus an unstable atherosclerotic plaque. So a stable atherosclerotic plaque is one that has a big fibrous cap, whereas an unstable atherosclerotic plaque is one that has a thin fibrous cap. So of the two components of an atherosclerotic plaque, we like the fibrous cap more than we like the lipid core. Atherosclerotic plaques which have very big lipid cores and very thin fibrous caps are at risk of rupturing, they're at risk of tearing. Okay, so basically what happens is a huge great chunk of the top of an atherosclerotic plaque will break off. Okay, and that's what we mean by rupturing or ulcerating of an atherosclerotic plaque. So unstable atherosclerotic plaques are at risk of rupturing. Okay, having a very thick fibrous cap puts you at much less risk of rupturing. So we like stable atherosclerotic plaques that are not at as high a risk of rupturing. We don't like unstable atherosclerotic plaques. Now, why do we not like unstable atherosclerotic plaques? Well, if an atherosclerotic plaque does rupture because it's got a very thin fibrous cap, what ends up happening is you establish the coagulation process on top of the ruptured surface of the atherosclerotic plaque. So remember, the coagulation process, coagulation means the conversion of the liquid blood to a solid mass. And coagulation is something that usually happens when you cut yourself. So if you get a hole in the side of your blood vessel, you want coagulation to occur because you want the blood to turn to a solid and plug the hole in the side of that blood vessel. Okay, now what activates the coagulation process? Well, it's tears in the wall of the blood vessel. Basically, the components of the blood, they usually have a very boring life. They only ever see the surface of the endothelial cells and other components of the blood. There are certain molecules that they are never allowed to see. So things like collagen, they're never allowed to see collagen. And when they do see collagen, it means that something is ghastly wrong. For instance, there's a hole in the side of the blood vessel, and they're now exposed to all the different layers of the blood vessel wall, many of which will contain collagen. So for instance, the basement membrane is full of collagen. Okay, they're usually never allowed to see that because the endothelial cells cover it up. Okay, whereas if you have a hole inside the blood vessel, they'll see all of these different molecules, and these different molecules that they're never usually allowed to see are what then trigger the coagulation process, this process where the blood turns from a liquid to a solid. If you get a ruptured atherosclerotic plaque, then you expose the contents of the blood to all of, well, loads of molecules that they shouldn't see, okay, and they interpret it as we're passing through a hole in the side of the blood vessel, we need to coagulate, and the coagulation process is activated. Okay, so you get a massive blood clot forming on the surface of the ruptured atherosclerotic plaque. Now, when the blood coagulates, the blood clots in the middle of a perfectly intact blood vessel, as it would be doing in this case of a ruptured atherosclerotic plaque, that is known as thrombosis, and the blood clot itself is called a thrombus. Okay, so those are two key words, thrombus and thrombosis. So thrombosis is the process of forming a blood clot, the process of coagulation occurring in the middle of an intact blood vessel, and the blood clot that you end up with is called a thrombus. Contrast this to the process of hemostasis, which means forming a blood clot in the hole of in the side of a blood vessel, and then the blood clot is called a hemostatic plug. Okay, now if you form a thrombus on top of a ruptured atherosclerotic plaque, that can completely shut off blood supply through that blood vessel because it can completely occlude the entire blood vessel. So the atherosclerotic plaque, you know, that was narrowing the blood vessel, but it wasn't completely obstructing blood flow. But once they rupture, they can then have thrombosis occurring on top of them, and the thrombus then can then completely shut off blood supply. Okay, and wherever this blood vessel was supplying is now going to become ischemic. And indeed, this is one of the ways that heart attacks, myocardial infarctions can occur. You have atherosclerotic plaques in your coronary arteries. That permanently lowers blood supply to the heart and gives you ischemic heart disease, for instance, angina. But then the reason it suddenly turns into a heart attack is that the atherosclerotic plaque ruptures, you get thrombosis occurring on top of the atherosclerotic plaque, and this then completely blocks all blood supply to that portion of myocardium supplied by that blood vessel, and therefore that portion of the heart dies. Okay, so unstable atherosclerotic plaques are dangerous because they can rupture and thrombosis can occur on top of them, and that completely occludes blood flow to the area that's supplied by the blood vessel. Stable atherosclerotic plaques, where the fibrous cap is much thicker, those are less likely to rupture, so those are safer than unstable atherosclerotic plaques. Okay, so we'll have a break here, and in the next video what we'll talk about is the anti-atherosclerotic drugs.